In our previous lesson from Chapter 4, we talked about what makes a cell, the different types of cells, and the internal structures of a eukaryotic cell. We're going to look at those internal structures in more detail here in our second part from Chapter 4. And we're going to start with the nucleus. And the nucleus is very obvious when we look inside a cell, but I do want to mention we get very used to thinking of the internal structures as flat, two-dimensional structures. We look at this underneath a microscope. This particular picture comes from an electron microscope. And the outside edge here of the nucleus just seems like kind of an oval. But keep in mind, this entire structure is three-dimensional. It's more spherical in structure. So sometimes in diagrams like the one over here, we can see they're attempting to show it its three-dimensional nature, even though under a microscope we tend to see a slice of it. So it's not really a flat structure. But inside the nucleus is the DNA, which remember is the instructions for how to make proteins. That's very crucial to a cell because cells are basically protein factories. Living things are primarily made of proteins for both our structures and our functioning molecules. So with those proteins being so important, that's why the nucleus ends up being so important. It's the library of information. Now, to protect that library, there is a double-layered membrane around it. So in the outer portion here, what's usually referred to as the nuclear envelope, it's not a single membrane like around the cell with the plasma membrane. It's a double-layered membrane. Each layer is extremely similar to the plasma membrane, but it's twice as thick to give a little extra protection to that DNA. But each layer is itself a bilayer of phospholipids, just like the plasma membrane is a bilayer of phospholipids. So actually, to get all the way through from the outside of the nucleus to the inside, you'd have to go through a total of 4 phospholipid layers to get there. The first bilayer, then the second bilayer. Inside the nucleus, you find the nucleolus. The nucleolus is where ribosomes are formed. Also extremely important because ribosomes are the structures that actually make the proteins. And if the nucleus is important for having the information, the ribosomes are just as important for having the ability to then translate that information into proteins themselves. The next area we're going to look at in the cell is what's called the cytomembrane system, or the cell's membrane system. Now, this isn't the plasma membrane. This is internal membranes. And we can see here we have some membranes that are here all kind of folded up and connected directly to the nuclear envelope. And that's very typical. We talked about the ER last time, and the ER actually is attached and kind of wrapped around. And again, to think of it in terms of the three-dimensional nature, this is almost like a big membrane bag that's all kind of wrinkled and folded up and packed inside the cell. But when we look underneath the microscope, what we see are just a series of lines. It looks almost like a maze. In fact, reticulum comes from a word that basically means maze-like. Well, the ER, like we said, it's more like sacs and tubes that are all kind of flattened out in there. And when we see that cross-section, that's where we just see the line structure. But these sacs and tubes are very important for helping the cell process proteins and lipids. We call the ER either rough ER or smooth ER, depending on whether or not it's covered in ribosomes. And that's not just a cosmetic difference. It's actually a functional difference. The portions of the ER that are covered with ribosomes are more involved in protein formation, and the ones that are not covered in ribosomes are more involved in modifying lipids. So, 2 different types of ER, 2 different functions, protein formation and lipid formation. Also part of the cytomembrane system are the Golgi bodies, and again, these are more like sacs more than anything else that are kind of flattened out. And here we're seeing the front edge still cut off to give us the inner view that we see when we look inside a microscope here. And coming off of these sacs are little vesicles, little spherical structures that are also made of membranes. So this is all membrane here. But more than anything else, I think the Golgi bodies look kind of like a stack of pancakes. And if you've ever made pancakes, you don't often get little drips that form mini pancakes. That's almost what the little vesicles are like. But they're actively forming off the edges of the Golgi. We call the process by which they do that budding. And the Golgi then is what takes proteins that were made by the ribosomes and probably modified by the endoplasmic reticulum and then processes them further, may add other structures to them, may combine a couple proteins together, or may add some lipids to the proteins, process these, and get them ready to become secretions for the cell. Now, some of those structures that bud off of the Golgi may actually contain digestive enzymes. If so, they would be called lysosomes. We mentioned before that these lysosomes have the ability to digest material inside the cell. That's not their only purpose. Yes, single-celled organisms like this amoeba here, the amoeba is wrapping a couple pseudopods around another cell as making a food vacuole inside the amoeba, and that food vacuole then will merge with some lysosomes to digest the food, and the amoeba can get its food that way. However, it's not just single-celled organisms. There are some multi-celled organisms where individual cells may also use their lysosomes for digestion. I think perhaps the most fascinating use of lysosomes, though, is if you have something like a frog here, or other amphibians that go through a metamorphosis. We all know tadpoles are baby frogs, and tadpoles have a tail, and adult frogs don't. Well, the tail doesn't fall off. Actually, cell by cell, lysosomes inside those cells burst themselves open and cause the death of that cell. The cell itself gets digested from the inside, and those materials then get absorbed into the body of the frog. So the tail doesn't fall off, it just becomes smaller and smaller as it is internally digested and able to be used for growth for the frog. Now, the mitochondria inside a cell are very important structure. Again, three-dimensionally, it's usually fairly oval in shape with these folded membranes, and we've cut into the diagram here to kind of see the internal structure. But when you look underneath a microscope, you're going to see usually a very flat-looking oval with little lines inside. Those lines are the inner membranes. Mitochondria are, of course, the organelles of aerobic respiration, and aerobic meaning with oxygen, the process by which ATP is produced using energy from food. An interesting thing about mitochondria is there's a lot of indication that perhaps they evolved from bacteria, maybe sometime back in the past, a very long time back in the past in terms of life on Earth, that some larger cells maybe kind of engulfed some smaller cells that were really good at processing food, and kind of created a symbiotic relationship where the smaller cells would process food for the larger cell would gain food for the smaller ones, and that trait has been passed down, and to this day, eukaryotic organisms have these mitochondria still inside our cells. There are several reasons why we think this may be the case. The fact that mitochondria have their own DNA separate from the nucleus, the fact that honestly they kind of look like bacteria, they have the similar shape, appearance, and size to that, but just also the fact that in trying to explain how eukaryotic cells might have evolved and come about from prokaryotic cells, it's just one idea that it's a possible way to get that internal structure that is so complex inside what was at the time probably a very simple cell. We call this theory the endosymbiotic theory, meaning internal, endo, and symbiotic for symbiosis, the two life forms living together. Well, inside each mitochondria, those inner membranes we talked about that are all kind of wrinkled, that creates extra surface area for the reactions to occur. Chemical reactions need a surface, and that inner folding allows more reaction to take place inside each mitochondria. Those inner membranes are referred to as the cristae. Another very fascinating organelle that we find in some plants, in plant cells, are plastids, and this is a colored electron micrograph of a particular plastid here, and they picked green for a reason. But plastids come in a couple different types. If it's a colored plastid, it's called a chromoplast. The prefix chromo means color. And the most famous chromoplasts definitely are the chloroplasts. The prefix chloro means green. Chlorine gas is a green gas, that's how it got its name also. Well, these plastids that are green, the chloroplasts, are of course the most famous ones, and their job is to perform photosynthesis. And you can see inside the chloroplast, it's a pretty complex structure. Some other chromoplasts you might have come across are ones that give the petals of flowers and the fruits of different plants their color. And so there are things called like rhodoplasts, which are the red ones that we can see here. Each of these little red plastids will give an overall red appearance to this part of the plant. This might be a portion of a petal from a flower that's red. And we find a lot of chromoplasts in flowers and fruits. There also are colorless plastids that are sometimes called amyloplasts or leucoplasts. They have two different names. The prefix leuco means without color. So leucoplasts, we've named them for what they look like. Or the other name, the more common name usually used, is amyloplast. Amylo comes from the word amylose, which is a type of starch. And colorless plastids do tend to store starch. We tend to find amyloplasts in things like the inside of a potato. These are actually some potato cells we're seeing here. The cell is the larger structure, and inside you can see those amyloplasts storing the starch. And so inside plants that store a lot of food, we'll find a lot of amyloplasts. Well, back to the chloroplasts. Inside a chloroplast, there's a lot of structures we said before. There are intermembrane discs, and each disc is called a granum. The plural of that is grana, so there are a lot of grana inside each chloroplast. And as you can see in the diagram, the label there, the overall term for all the grana is usually referred to as the thylakoids. And then surrounding the thylakoids, surrounding those grana, is a liquid called the stroma. And when we talk about photosynthesis in a few weeks, we'll learn about what takes place in each location there. But again, it's all about surface area and kind of compartmentalizing so that the chemical reactions of photosynthesis can take place better. But inside the grana, that's where we find pigments. Pigments are what we usually think of as colored chemicals, and that includes the colored chemical chlorophyll. So the organelle is the chloroplast. Inside the inner membranes of the chloroplast, we find the chemical chlorophyll. And just like mitochondria, there's a lot of indication that perhaps chloroplasts also evolved from bacteria. Again, they look like them. They act almost like they have their own functioning, and they have their own DNA, just like the mitochondria do, separate from the nuclear DNA. Cells have a cytoskeleton. The cytoskeleton is primarily these tube-like structures that we see kind of stretching through in this diagram, the long, thin tubes and filaments. The main difference between a microtubule and a microfilament: microtubules are more rigid, microfilaments are usually more flexible. You can think of the microtubules kind of like the bones and the microfilaments kind of like the muscles for the cell. But in both cases, these are very tiny, very thin structures. They're difficult to see. This isn't a photograph here. This is a computer simulation showing the structures. They're very difficult to really see in most photographs from the inside of cells. Now, a weird thing about the cytoskeleton that makes it different from our skeleton is even though some parts are permanent, a lot of parts are temporary. Some of the portions only form when they're needed, and then when they're done, they kind of fall back apart again, almost break back down, which is obviously very different from our skeleton. Some of the more famous parts of the cytoskeleton, not found in all cells but found in some, are cilia and flagella. These are on the outer surface of a cell. On the left here, these are typical of some flagella here. We often think of a cell with one single tail, like you might find in a sperm cell with the flagella. But often there may be several. There may be flagella at each end, or there may be lots of flagella kind of all over the surface of the cell. However, these all are very likely to be involved in moving the cell around. Ironically enough, even though we picture this as a tail, most often a flagellum actually is in the front of the cell and kind of pulls the cell from the front. But flagella in general are long and fairly few in number, as opposed to cilia, which we see here. This is an external picture from a scanning electron microscope of the outside of a paramecium. And you can see it almost looks hairy. And each one of those tiny little hair-like structures is a cilia. Well, both cilia and flagella are basically the same structure. They're both microtubules, very tiny little tubes on the outer surface of the cell, but they're both involved in motion. Inside the cell, some other microtubules we find. Centrioles are a fairly famous one. Centrioles are these two little bundles of tubes. We've got some in one group here, and then a second cluster that runs kind of in right angles to it. They're kind of hard to see here inside the diagram. But there are these two bundles that are involved in animal cell division. We only find the centrioles in animal cells, and they're found in a region of the cell. This whole region is referred to as the centrosome. But they're a special type of a microtubule called a microtubule organizing center, an MTOC or an MTOC. And these MTOCs seem to somehow organize the processes of division for the animal cell. There are other MTOCs in some cells that help the cell do some other functions. One interesting thing we find in a lot of these microtubule structures, including cilia and flagella, and including often the centrioles, is a very special arrangement. If you look at, we're looking at a cross cut across a cilia here, a cilium. And if we count, we've got 1, 2, 3, 4, 5, 6, 7, 8, 9 outer tubes and one set of inner tubes that has two distinct separate portions. We usually refer to the structure of these microtubules, including cilia and flagella, as having a 9+2 array. Structurally, they're made of 9 smaller tubes around the outside and 2 small tubes in the middle to give a little bit of strength and support there. And those tubes are on the full length. So again, we're looking at the cross section. This would be kind of like if you would cut across a pencil and you'd see the lead showing on the inside. That's what we're looking at here with a microtubule. Cell walls. As we said before, cell walls are actually typical of cells. Most cells have cell walls. There are some protists that don't. There are a few unusual cells here and there that don't, but no animal cells have cell walls. The best way to think of a cell wall is really not a part of the living cell itself. It's actually just a little box that is around the outside of the cell that is what the cell lives inside of. These are some plant cells, and the internal living structure here is surrounded by the cell wall. The cell walls divide one cell from another. They give the cell shape and support. This is often what helps plants to have the rigid shape they have and not just fall down. Plants don't have skeletons like us in many cases. The cell wall has to have the ability for things to get in and out of the cell, though, so there have to be holes. Those holes are called pores. We're looking at an electron micrograph picture here of some pores in a cell wall, and those are large enough that even good-sized molecules can pass through these pores. They're much larger than the holes that we would have in a cell membrane to allow things to go through, which we'll talk about those a little bit more in Chapter 5. Now, all cell walls are made of polysaccharides. It's different polysaccharides. As we talked about in Chapter 3, you can have something like cellulose in a plant cell, and that makes up those cell walls, or maybe chitin if it's a fungus that we're talking about. They're both polysaccharides. They're chemically different, and they're found on different types of cells. Those are some of the key structures we find in eukaryotic cells. As always, I would strongly recommend you read through your chapter, especially sections that you didn't understand or you felt we went through a little too quickly here. And as you read through in Chapter 4, make a special note of anything you want to ask about, and definitely bring your questions to class so we can talk about them. And everybody have a great day.